Today it's about structure comparison. Again, I believe that all email communications should go through Lothar. The man behind the camera is Jonas Rape. Some of you have heard Tim asking for Max before. So last week you had a recap on the constituents of a protein structure. Today we look at structure comparison, and next week we have a recap on alignments. Let's begin again with the other idea of units, namely the idea of domains. Proteins are made of domains. Where are they? Can you see the domain? Domains. Where would you?
Yeah, with the right one, I would say the small part on the left is probably one domain, and yeah, the big one is at the one on the right maybe is another one.
So you cut here. So is that what? Is that? No, I'm asking you. Oh, sorry.
I was with the right one.
Yeah, on the right one, I believe. So right one, you do like this. Everybody agrees with that? Do you cut here or do you don't cut? Oops. Let's do it differently. So you obviously have both options. You already see one way, in fact, of assigning domains. I'm not sure why the size is not quite fitting. I have to work on that. But so one way of, in fact, once you see the three-dimensional structure, identifying domains, look for clefts, look for holes, look for low density, and cut. That's essentially what you visually did, right? Now, here's another point that this slide sort of indicates, which in fact, if you sort of turn it around a little bit, then you could possibly see that, in fact, this unit is repeated here. It's a different scale. That's why it's difficult to see, and it's a slightly different orientation. So that then would suggest to you, maybe there's a cut here, although you may not clearly see this lower density. But there's something else that you see here that sort of could indicate a cut.
With the helix and patterns.
Yeah, you see, this is sort of looks all alpha, this sort of looks mixed, but this somehow, in that sense, just in terms of secondary structure composition, looks different, right? Maybe it's a wild cut, but that sort of gives away that there is something already that might be different. Now here's the other point. It's seen again. And that's another theme for, in fact, cutting domains. Something that you see in different constructs, in different proteins, in slightly different ways. Let's just get a few of the simple. So Andrea told me that you knew everything she did last week, or whoever was here. But let's still have some quick recap on a few issues. So the statement is, longer proteins are rarer than shorter ones. Is that statement true? Anybody feels the statement is wrong? No. Prokaryotic proteins, so I'm going to use the acronym here, totally misleading. Procas as prokaryotic proteins, and eucas as eukaryotic proteins. Procas are shorter than eucas. Anybody disagrees with that? At least one person in the room disagrees, and that is me. Not really. I'm going to show you data on that in a minute. Some proteins are multi-domain proteins. Most are not. Nobody knows quite to agree on this. So I believe I see a question mark. Does anybody fall either way so far? Everybody's face I see has a sort of question mark written over it. Not really. In fact, most proteins are multi-domain, and I'll show you a figure about that. There are more proteins with 3 domains than with 1. No, I believe that your smile was a no. Or,
I'm going for yes.
No. Oh, you're going for yes. I meant. Oh, I forgot to print out the photo, the names. Your smile was.
Yeah. No, I was just thinking about, because the one before says most of.
Yeah. Thank you.
but
let's just see how aware you guys are in 2 months from now. This is an amazing statement that you smile about that. Maybe this is because it's spring and all your energies are there. Let's see towards the end of the semester. You will not be as fit anymore. Point taken. So there was one no, and it's about true, and I again show you data. Do prokaryotes have more domains than eukaryotes? So there's a no. Is there any yes? In fact, I should have asked this question the other way around. Typically, it has been assumed that eukaryotes have more domains than prokaryotes, and that, in fact, is also not true. What is true is that, I'll show you again the data in a minute, that many eukaryotes, or eukaryotes have many more proteins with many domains. So here are two views. Green, percentage of all the proteins on the axis here. Green, PDB, proteins of known structure. Blue, entire proteomes. In this particular case, 62, a little bit outdated data. Number of domains. First, you see that clearly most proteins for which we know the structure, 60% have only a single domain. While the blue suggests that number of 3 domains is almost similar to with 1 domain, and there's something more to be said that I will not do now, but since this is less than 30%, this clearly is less than the majority. So most proteins have more than 1 domain. Okay? Any clue to why the green is different than the blue?
1 domain, or PDB, is embraced for small globular protein.
Why?
Because there is a crystalline.
Exactly. That's totally the bias that you see. And that bias, in fact, has given most people this idea that most proteins are single domain. And there's another bias, in fact, the experimental bias. Proteins with 1 domain, shorter proteins, are also easier to handle in other experiments, not only for crystallography. Any kind of molecular biology is easier done with proteins that are shorter. So we have, in the annotations, we have a lot of overrepresentation of simpler proteins, and single domain proteins are simpler. Okay? So most likely this blue, in fact, underestimates the reality. So most likely this will even go down, and will in fact go down to the level that 3-domain proteins are more prominent than single. This one is a little bit more complicated. What it shows is the number of domains in a protein versus the length. First thing you see, blue is eukaryotes. This is essentially, in red, is essentially prokaryotes, and that's the PDB. You see they're all straight lines. That implies that essentially all domains have a similar length. And the average domain length, essentially the slope of the line, is about 100. The curves, number 1 here, is higher than 100. And it's in fact differentially higher for eukaryotes and prokaryotes. So there's a difference. In some sense, this suggests that there is one long domain, and then you add others onto it. Okay? That's like an enzymatic domain that does something, and then you have a couple of SH2, SH3 domains that steer, that do ATP, that have something to do with how this protein is regulated, how this protein is in fact interacting in the machinery. So that explains why most protein domains are about 100 residue, but the first one is longer. Okay? But you already see that in fact prokaryotes and eukaryotes have a similar length in their domain. Here, let's get back to the length distribution. Blue, eukaryotes; green, prokaryotes; archaea, misrepresented. In some sense, the peak here is explained by the fact that this is a relatively small data set. So that was drawn at a time when we had 60 entirely sequenced organisms. Now we have thousands, many thousands, so we would have to redo this curve at some point. But you clearly see that the average in the blue, again the blue here was done on 5 entirely sequenced organisms, essentially is very similar to the green. You also see that there is such a wide spread in terms of the length. Here we go from something that is in the typical domain length of 100 residues to something that is 400, and that's the ballpark of the distribution. So to say that a protein is 230 residues doesn't make sense. Okay? That's another thing that is very important in this raw data. There is no reasonable answer to the question how long is an average domain. You can put down a number, but actually, given these distributions, doesn't really make sense. This curve here shows essentially the same thing, prokaryotes, archaea, but is a cumulative distribution going on this side here and on. And what you see, so while largely the eukaryotic distribution is very, very similar to the prokaryotic distribution, eukaryotes clearly differ in the fact that they have many very long proteins. So 1,000 and longer. Those you significantly more often find in eukaryotes, and again, many of those are long fibers coil based. Any idea what the longest protein is?
Actin filament?
Actin filaments. Actin filament actually are not really a single protein. They are, in fact, this motif of a repetition of a domain, or a repetition. Actin filaments, in fact, are repetitions of protein units, or proteins as unit chains, even. But it's a good guess. In fact, actin filaments are sort of the longest aggregation that we typically know of a protein that is sort of visible. Hair is another example of that. Nerve. No, but there's something that in fact is part also of the actin filaments, but it's something really big. Any idea what the number is? So I show you here, there's a distribution until 2000, so it must be clearly longer than 2000, right? That much we know. How much? If I said 5, would anybody say no, that's too much? No. 10, that's too much. 30,000, Titan. Titan has 30,000 residues. It is in fact true, we do know at this point just about a handful of proteins that are longer than 10,000, and Titan by far takes the cake. 35,000 residues, one particular protein. Okay, another set of issues. So here, I believe the answer is very trivial. How many proteins in human, you know, is 20 to 25,000 roughly, and the number goes down over the year. How long are they? And I already said it's from 35 to 30K, and the median is around 400. Doesn't make sense to use that. They consist of units. They have more than 2 domains. How many proteins are known? Oops. Who didn't read it? No, shoot. No, okay. I'm sorry. I'm really sorry. So it's 52 million at this point in UniProt. And so let me ask a different question. So 52 million. Oh, the other question, it doubles every 18 months, which in fact is fast. It used to be, until 98, people said that computer power is doubling every 18 months. Then the number was revised, so the duplication of computer speed, disk price, computer price, or whatever you measure, is less than every 18 months. It was more than every 18. Doubling takes now 24 months. What about the biodata? Is that slowing down? So the last duplication from 26 to 52 took 18 months. How long was the one from 13 to 26?
I guess much longer.
That is a question of...
10 years maybe.
Oh, no. At some point in time, your answer would have been right. Now, these days, before it was 22 or 21 months. But then this duplication here decreases. So not only is, in fact, the biodata growing faster than the computer, but in fact, the growth rate is also higher. There's another problem for computational biology every day, and that in the next lecture we are going to talk about alignments. So what's the problem in this number?
So you have to align them against each other.
Essentially, it's a square. It's not only that the biodata is growing faster, but you have to square the problem. And that, in fact, is where the problem becomes a nightmare. And that is an everyday nightmare. Prokaryotes and eukaryotes obviously use the same amino acids, but do they use the same distribution of amino acids? Oh, I forgot. Why do you answer that? I forgot to put the slide in for that one. Yes, they do in fact use essentially the same distribution. Nevertheless, so by the way, this is selenocysteine and cysteine. You guys are aware of that? Selenocysteine, in fact, is a derivative of cysteine. That cysteine has a sulfur here. That's a selene here, and that in fact is only in a tiny percentage in eukaryotes. So that is the 21st amino acid that does not exist in prokaryotes. That is a trivial, I call it trivial because it doesn't really, arguably doesn't really matter a lot. So there are 20 amino acids and they also have very, very similar biochemical features. But essentially, in fact, the amino acid distribution between all prokaryotes, all eukaryotes is almost identical. Sorry, I owe you the slide, I'll show you again. Nevertheless, you can in fact predict the organism, and that's the paper here from Itzhak Peah in the group of Beckman, published 10 years ago, in which they trained machine learning devices to predict if they get the entire amino acid composition of one organism, they sort of can predict the organism. This is surprising because the overall level is similar. Nevertheless, particular organisms have particular biases, and that has to do with the features of DNA. It has to do with a variety of the environment in which the organism lives that may in fact imprint this changes. So you do know that in 95 we had the first entire sequence organism sequenced. The first organism entirely sequenced. Haemophilus influenza, the first eukaryote was in 96, was yeast, and many others followed. Now, we have 52 million sequences. How many do you believe of those come from sequencing entire organisms? Most? No, it's not most. That's true. Any? Throw a number at me.
10%.
That's a very good number. It's about between 15 and 20 million. So the number, however, is increasing. That 52 million is not counting the largest repository of data that we have, which is metagenomics. You know what metagenomics, the idea is?
Where you sequence the whole community of bacteria? Community,
now this is this one way. So the initial attempts at metagenomics, in fact, from the Claire Fraser in the 97, she started that. They dug from the soil in Minnesota. They took it out from the air in Manhattan. They take it from the ocean, so there's the yacht going all over the planet and just diving in, taking out all the material. So those are the first attempts at metagenomics, and then you essentially sequence whatever you get, which in fact is a symbiosis, is a system, but not in the sense of the gut in the way we understand it today. The idea really is environment. The idea is the whole thing, but that really was also to just create a flash view at something that we don't really classify. The impression is from the sequencing that there's a lot of things out there that we've never seen before. But the problem with shotgun sequencing is, so you go into the soil, pick out a little bit, and it's full of things that you haven't seen before, but you actually don't know what the organism is. And that goes back to this one. So in order to predict the organism in this paper here, that was simply establishing that the amino acid differences are significant enough to use them for a predictive purpose. But there's no biology underneath. It's just showing that they really differ. Because we need, for this prediction method from Itzhak Peah, we in fact need essentially the entire amino acid composition of the entire organism. When I pick this piece of a soil, I have some small fraction of that particular organism. And from that, in fact, I cannot predict the organism. And most of these organisms appear to be not known anyway. The reason why these data are not in the 52 is because these data are full of errors. And the database providers, Uniprot in this particular case, refuse to just dump them in full scale. The number of those metagenomic sequences is much depends a little bit on how you look now at the data, but is much larger than 52 million. But that is not publicly available, or it's not available through UniProt. It's not in the 52 million, because essentially to identify what a protein is the problem. So the strongest data is where you have so many experimental evidences for one particular interpretation. This is a protein, and of this strongest you have a few million. Of the weakest you have in the ballpark of 500 million. Anyway, okay. So how would you predict a domain from sequence? Let's just go forward and backward. I will present these numbers again to you, and but still, so I said that there are about 52 million sequences known. How many proteins do we know the structure for in the PDB? So the one that is biased by single domain proteins I showed you. Ballpark. 10,000, 100,000, 1 million?
In the thousands, 60,000?
Yeah, that's a very good number. So ballpark 60,000, 100,000, depends a little bit on how you count. Proteins at this point I believe is 75,000. Now, 75,000 structures, 52 million sequences. Okay? So for most domain assignments, again, the idea, one idea that you had, that everybody who sees this picture immediately jumps at, is you look at the three-dimensional shape and you look for density, for local density. That's the way you define domains. And again, today is about comparison, so finding the similarity between these two domains. But for most of the proteins you obviously don't have the structure. How can you identify domains?
One possibility would maybe be to do secondary structure prediction of the sequence, and then try to, or you could just, because I think the number of domains is much smaller than the number of proteins, you could try something like homology modeling and just try to guess the overall structure from a sequence and then try to do the name.
Does everybody in the room know what homology modeling is? I take that as a yes. How many proteins can you build models for?
Well, everything that is homologous to those 70,000.
Yeah, how many are those?
I'm gonna guess not all the 52 million.
Safe.
I would, to get the domains, I would just, maybe you just look for sequences which repeat in the proteins, like not 100%, but I don't know, 80% identity or something.
Yes, that's great. So we have two ideas now, homology modeling. So we look for things where we in fact can map it to the known 3D structure, and then we go back to the problem that I showed before, density, which again, I may come back to, is not totally solved, but the principle is clear. The other idea is we look for repeated patterns. Any other idea? Secondary structure was another idea. Oh, there was somebody else, or was that also you with the homology? Yeah, you started with secondary structure and then you got into homology modeling. Okay. Yeah?
Would we try machine learning? Like training an algorithm on proteins of known domain structure and then just trying it on everything else?
Yeah, we could.
Does it work?
Depends on who you ask. So there's a method called SHOPNet, and we have been working for many years, and we have published many papers in which we claim that we are the best, but I will never talk about it. So in between these statements and not statements, you sort of get an idea, but I'm not sure about the answer to your question. And maybe in the lecture on the challenges for experimental structure determination, I will get back to it. Essentially, yes, it works to some extent, but the question is it useful? And that is the challenge. Okay, now here's what I do. I have a sequence indicated by this long bar here. I run it against everything in the PDB for which I have a domain assignment. I find this domain assignment and I cut. Okay? I cut that that is a structural domain, so I say this is a domain. This fragment here is too short, so I throw away. What would you call too short?
Less than 50?
That's a great number. Less than 50, less than 20 or 30, wherever you go. So where you can be sure this is not going to be a functional thing, right? It may be an additional end. It may be a signal peptide that directs you where you wander in your cell, in your native protein, but it is not really a structural region. The next idea is the one that essentially, in some sense, you brought up, and we will get back to it in more detail. PFAM, you have most likely heard, is the protein family database. And the protein family database essentially is exactly a manually curated, expert curated collection of regions where you see repetition. So you start with a protein, you find everything in which you have some similarity, and if you find a lot and you look at the annotations, as in what did people do experiments, what did they find, what similarity do they find, and then that is annotated as a PFAM family, as a region that is like a domain. Okay? And you see that, you cut again. In this particular case, we don't fall below the 50 cutoff, so now we have essentially two domains assigned, one fragment that we throw away, and two fragments that remain to be assigned somehow. And that now is essentially a similar idea, but now we in fact look SwissProt. SwissProt is a database of proteins for which we have some experimental information. You are deposited in SwissProt when an experimentalist has worked on a protein, so we already know this is for sure a protein. When I said we know 52 million proteins, then they may not be proteins. Those are gene products. Those are things that we assume form proteins, but that may be wrong. For the ones in SwissProt, which currently are in the ballpark of 400,000, we in fact know because somebody has in fact put that construct into a cell, has purified that protein, and has done an experiment with that protein, and there's a report about the outcome of that experiment in SwissProt. SwissProt is in fact reducing its size over the last few years because they make an attempt to throw out annotations that are not reliable. Okay, now assume you have in SwissProt 1, 2, 3, 4, 5 proteins, possibly in different organisms, and that's just the way they align. And you see, looking at these 5, you may immediately suspect, okay, we have two different domains here. This is shared by 2 proteins, this is shared by 2 proteins, and in fact this one here has both domains. This implies that we can cut again, right? We have a clear case here to cut this fragment into 2. The other one we can't do anything because we don't find any further reason to cut in today's databases. That then may become a reason for machine learning based assignments of domains, and we have, in this particular case, we would argue that this protein has 5 domains. And we essentially did something like that for the target selection in structural genomics. Okay, now again, many of the slides that I show today go back to something that Marco Punta, who's now at PFAM, has given. Let's begin with words. Protein structure in 1D, 2D, 3D. So 3D is the 3-dimensional shape. It's the coordinates. Okay? What is 1D? Or what, let's do it the other way around. Where do you see 2D in this plot? Yeah, I'm not giving it away. And these are just, to repeat this part of Andreas' talk, different views of, in this particular case here, the potassium channel. This is the molecular view, just a sec. This is the rod view where the helices are cylinders. There's a beta strand here, a porin. Yeah?
So the 2-dimensional structure is the secondary structure, so you'd see there.
Why would you call secondary structure 2D?
Well, because it's the next level of organizations if we assign 1D to the amino acids.
Yes, you're very right. This is, in fact, historically the way many people use these terms. They invented 4D, that's very intuitive, which is sort of the way domains fit together or proteins fit together. And maybe then there's a 5D and a 6D, and it begins to become increasingly intuitive, because we're all very good at imagining 6D objects and comparing them. On a physical level, I'd argue 3D is very clear, 3-dimensional, right? 2D is 2D information. We are computer scientists after all, or we have at least some solid footing in computer science. And a 2D sense is a contact map. So what I plot here is the residues down here, the residues down here, and every entry here is, for instance, an energy of binding between two residues, or a contact map. In this particular case, a distance map. So where this is the energy scale is directly proportional to the distance according to some simple energy assumptions. So the darker, the closer. Trivially, the diagonal peptide bond is black, and then you see sort of some pattern forming. And the pattern formation, in fact, reflects the secondary structure units, but 2D. It's 2D to write it down in 2 dimensions is a matrix. 1D is a string. 1D is a sequence. 1D is secondary structure, because for every single residue you have an assignment of the secondary structure. You know it's an alpha helix, a beta strand. For every single residue, you know its solvent accessibility. You know where the protein is on the surface or not. It can access so much water, but it's a single number. It's a 1D feature. So I prefer to think about it in this term, because that essentially is the information content of the information I have. And I will continue to use these terms and in fact present also the lecture in terms of 1D, 2D, 3D. So prediction methods in 1D, 2D, and 3D directly. 3D classifications. So essentially, if you assume this is the structure universe, different bubbles here are different islands, and there is this enterprise of structural genomics, and the idea of structural genomics was green islands are those for which we know experimental structures. White ones, those for which we do not know the structures. So the idea of structural genomics initially was, let's just have a large-scale effort defined as $180 million per year for 15 years to fill the white islands. Okay, that was the initial concept. Now, do you have any issue with this plot? So again, the idea is that an island is a structure. So all the proteins that are similar in terms of their 3-dimensional structure to something that is populated here on this island. What I mean here, that some of these islands are a little bit green and a little bit white. What I mean is there are some additional structural features and they are not known. So we know maybe the structure for the core, but there are embellishments, there are things put around it. And for those, maybe we don't know the structure. That's what I meant. But other than that, is there anything that clashes with you, yeah?
There's again this thing that proteins are made out of domains.
Yes.
And so proteins that do not really belong to the same island might share domains.
That's correct. So this island idea, so that's the first statement. This island, thinking about this island idea only makes sense if you talk about single domains. So this would be a comparison of all domains against each other. Is there another way that, another word that you have heard that would come to your mind with this picture? Have you heard the term fold? That's another word that is not domain. You have to understand, like many words in biology, is absolutely not well defined. We talked about visually cutting. We talked about evolutionary cutting. We talked about all kinds of, it could be that, it could be that. But there is no clear definition of domain, okay? We can define domain as minimal contacts within each other or something. But that, in fact, does not completely reflect the biological reality that we see. Still, here, what we compare are single domain and typically what people call folds. But it's a construct. Again, what's the difference between a fold and a domain? Typically, it's the same thing. Because the idea of a domain in structural biology typically is you throw this thing into water, it adopts a unique three-dimensional structure on its own. And that typically is assumed to be a fold, a single domain. So again, that is underneath any other issue that anybody sees. So the underlying issue here is in fact utterly not contemporary. This is a picture, comes from 98. It's totally outdated. It's one way, and this is very often happening in biology, is one way of thinking about it. There are folds. There are databases, I will get back to that in today's lecture, that are built upon this idea that there are folds, that there are different domains, there are different islands. But clearly, the water level between these islands is very shallow, which implies that we can actually sort of walk from island to island, at least for some. We do believe that there are islands that are sort of separated and cannot easily be reached. But whether that is true or not, we don't know. Ultimately, you know that all proteins evolved from some common ancestor. So maybe, actually, that all is sort of just an idea. And all, at the end of the day, you could argue there are two classes, the alpha helices, beta strands. And everything falls into these two classes. That would be absolutely simplifying, and it doesn't work. The reason people tried this classification, the reason why it doesn't work is it doesn't relate to function. You want a classification of structure that tells you something about function. But there's an issue with that, okay? Now, the goal is that, in fact, again, I said it, from the similarity in the three-dimensional structure, you want to somehow infer similarity in function. And you want to learn from the 3D structure. By knowing the 3D structure, you hope that you learn something from the function. And you actually want to learn something about evolution, in the sense that maybe you cannot identify the relation in terms of sequence. You said immediately 80%, so they have to be similar enough so that I don't make a mistake. When on top you have a structure, you have an additional identification that they may be related, and maybe you can go back further in the evolutionary time. And you want to use all of that to classify. Here's an example. And I somehow dropped all my references. So that is from, the next one has dropped too. Christine Orengo's lab, CATH. Both views here. And simply, you have, again, two different representations. The point here is you see the same thing with different, Christine calls it embellishments, different things around it. Christine and Janet Thornton coined this term. Oh yeah, here is her name. Here we go a level further down. There we go into, you mentioned secondary structure, so you now see repetitions of units. All you see here is the secondary structure elements put onto a string, and somehow, that was your idea before you got into the homology modeling, you're right. So immediately something jumps at you by looking at it. You still don't quite know the 2D reality, how this, whether the beta strands here are below or above the helix, how they are wound, whether they're parallel or anti-parallel. This is also important, and that is not visible here, so that's a 2D information. But in principle, the point is that you have repetitions of the same basic unit. And that, in fact, again, is the idea of a fold. Historically, the way they got to this, and the CATH database, Christine Orengo and Janet Thornton, was one of the first, was simply to see that some structures, some units, some motifs, some three-dimensional objects have been observed more often than others. Okay? So there must be some limited number of shapes. And you said that. The number, was it you? Somebody said the number of domains is limited in this? Or was it a different group? But still, fold remains an assumption. Again, in the most extreme way, and that was introduced by Michael Levitt here, the all alpha, all beta, alpha, beta, so secondary structure similarity. Now, we want to recognize the similarity. And there are three different ways of doing, or three different main databases of doing it that I want to present here. There are many others, but I want to focus on those three. SCOP, CATH, and COPS. QSCOP top mesh. This is a bunch of algorithms here. So let's begin with SCOP. SCOP is done by Alexei Murzin, and Murzin is one of those incredible people. And in this particular case, so Murzin, that is done in 2011. 90 publications is a very impressive number, but not that much. One with over 3,000 quotes, that's a very impressive number, and you will not find many at the TUM who can state that. 13 over 100 quotes, again, that's an impressive number, but it's not just, out there on the scale. The H index is impressive, but not that high. Murzin is the only person in the world who can look, could look until a few years ago, could look at a structure and say, I have seen that before. Until the protein database, I can't remember exactly historically how far he got it, but certainly until it had 20,000 structures. He just could look at a picture, or at least you gave him the computer file of your structure, and he could say, I have seen that. Sometimes he needed a little bit longer. He jumped at it, I have seen that, oh, I was wrong. But I've never seen him take more than half an hour on this. Because essentially he memorized every single protein structure in his head. Okay? And that's nowhere written here. And there's one person left. So Murzin started SCOP in 1993. At that point, I know at least 4 people who still could sort of do something like that. But very soon thereafter, he was the only one. And is an immense talent he has. Alexei Murzin is Russian. And he argues that he had to become that way because in Russia they couldn't afford the computers and he had to memorize it. Be that as it may, he's just gifted. So his idea was to build a hierarchy, and again, the hierarchy started from this old concept of all alpha, all beta, all alpha, beta. In the example here, all alpha, so you begin with a class. The class is the alpha, beta, and so forth, and you have some sort of other constructs. Now, we get immediately by reading through this. So until here, you may in fact say, okay, that is sort of secondary structure based and is trivial. But then you begin to see things that small proteins. So why would that be an own class? And now this is a biologist at work. So this is not a physicist. Alexei Murzin is a physicist in background, but it's much more the biology thinking. And the biology thinking essentially comes out of the fact that I classify first like this, and then I come to my limits, and I open a new class, and it continues going like this. 2 very often observed folds, the so-called Rossmann fold, and the Tim barrel, that's an enzyme here, where you in fact always have the alpha, beta, alpha, beta, alpha, beta unit repeated in a ring-like structure. Now, Alexei Murzin, by the way, continues to feel that there are folds and that his classification in many ways is real. The fold definition comes same major secondary structure in the same arrangement with the same topological connections. So topological connections means essentially is the beta strand below, in the one that I showed you, below or above the alpha helix. And how are they connected to each other? So are they parallel strands, for instance? Peripheral elements may differ. That again is the idea that I introduced before, embellishment. So things that you somehow put on a terminus before, after. That is additional, it may not be counted here. If now comes a new definition, 50% is accepted. So you have 50% additions. As long as you recognize half of this thing as the core, this is still fine. And then here's another point. The evolutionary relationship is unclear. So maybe I should have turned it upside down. I'll get back. As long as the evolutionary relationship is clear, he calls it a family. So if he loses that and only finds out that there is some relation through the structure, that's the part that he calls the SCOP fold. And those are in fact 3 different folds of the class alpha beta. They're all Tim barrels. Now in the SCOP hierarchy on the next level comes the superfamily. Anybody in the room knows what the idea of a superfamily? Originally actually was introduced for CATH. I will go next to CATH by Janet Thornton, and I'm not sure who actually coined the term. Maybe Janet Thornton, maybe Christina Ranger, I don't know. I don't believe it was Alexei Murzin. Actually, maybe it was Cyrus Roth. Anyway, sorry, I have to look up the history of that. Yeah?
So I think these are proteins of the same fold that share a function, but not necessarily an evolutionary origin.
Yeah, very good. Probable. Actually, he believes. So that's now we get into interpretation. So those are cases where Alexei Murzin believes they are evolutionary related, but it's not trivial to see. So you're totally right on that. It's not. You look at this. You need a Murzin to identify it. It's one way of defining it. It's very low. They share the same fold, that's what you said. They have a similar function, and essentially Alexei says that he can recognize reason why they are related. Whenever you have seen him do his magic in real life, you will be somehow convinced that he can trust him. And then we get to the level of family, and that in fact is the sequence based. So now we have a clear evolutionary relation, we have a high level of sequence identity, but not always are families defined. For instance, in globins it can go down to 15% sequence identity. That's SCOP. SCOP clearly is one of the most used databases of protein structure classification. The problem is you always have to ask Murzin, Alexei Murzin, if you want to know whether your protein is new. Okay, it's all in the head of one guy ultimately. SCOP is brought to you by a larger group, and there's a lot of automation now going on, but the ultimate decision is still a judgment call. So here's the approach: classify protein structure, CATH, from Christine Orengo, here in front of the Table Mountain, now at UCL, University College London. She's a CATH possessor of 120 publications, 1 over 1,500 quotes, 16 over 100 quotes, an H index of 40, very high. One of her first tools that she developed for a doctoral degree with Willie Taylor, in fact, was SAP, and that, in fact, is a method that compares structures. So how can I find out whether protein 1 or 2 proteins are similar to each other? And that led to the development or classification of a large database, CATH, and she did that together with Dame Janet Thornton, who is the current director of the EBI, the European Bioinformatics Institute in Kingston, Cambridgeshire, England. She has a bachelor in physics, Nottingham, a master biophysics, did her PhD in biophysics at UCL, University College London, was many times listed by various public magazines, Time and the like, as top 100 scientists in the UK, over 400 publications, 1 with over 11,000 quotes, 7 with over 1,000, 81 with over 100, H index of 88, a giant in the field. Which in fact, by the way, you don't often see directors having a record like that. Janet Thornton, she will, she will leave the EBI next year. And for me, that was sort of one of the most impressive pictures in their first publication of CATH in '97, a universe of protein structures. So simply to classify and put down, again, this is a long time ago, you could no longer do anything like that today, but in '97, this was the entire gallery of the different folds, the different protein structures out there. Okay, and that was a method that in fact got to this slide. They got to this slide through the principle class, architecture, topology, homology. Homology, okay, let's begin here. And I believe, no, I'm going to do it top down. Let's talk about homology later. Class. So class again is the same idea. Alpha, beta, alpha, beta in some sense. Mostly alpha, beta. Architecture is now a classification according to your overall shape. Now, in architecture, you still ignore connectivity. So you compare these helices, the rods, but you remove the loops. Don't worry how the direction goes, okay? In topology, you bring that and the connectivity back. Here, you look at the evolutionary relation. In 2011, the numbers are higher. We had 1,200 folds, 2,500 superfamilies, 11,000 sequence families, and 24,000 domains. So the way CATH finds domains has, in fact, the whole CATH classification is built upon domains. So the first step is really find a domain, and find a domain is a repetitive three-dimensional unit. Now, the way they do that is they use 3 different domain finders. What the domain finders do is essentially exactly what you do. They find local density. 3 different algorithms. When these 3 find the same solution, which in fact is only for 20% of the cases, it's fully automated. When they don't, there is additional work involved. And in fact, they then look at additional matching and ultimately look at the structure. But in contrast to SCOP, there's at least some attempt here to do as much as you can with automated methods. Okay? Now, from the domain to the superfamily is you now find this motif in some sense the same way you said it in terms of sequence, 80% again and again. Can I find it again? Here is the same motif but in 3D. Okay? This one is an SH3 domain. You look at the repetition of that domain and you build up your superfamily, your family of things that are related. Here is the role motif of the SH3. Now, there are some thresholds, more or less arbitrarily defined, when you call things same domain, same superfamily, not. And you sort of build up one by one these large CATH families. Here is a method, a set of methods that initially was called COPS, QSCOP, and that is the quantitative SCOP. COPS, can't remember that, the classification of protein structures. Top match has a meaning too. Underneath is Manfred Zippel, came at the University of Salzburg. He's also CASP assessor. Oh, I have said that 3 times now. Do you all know what CASP is? CASP is the critical assessment of structure prediction. And it's just a competition. I'll possibly get back into that. It was started by John Molt in 2004, I believe. 1994, I'm sorry. In 1994. And the idea, so the problem underneath was, let me go a step back. Can we predict protein structures? So I take a sequence, I throw it into water, it adopts a unique 3-dimensional structure. All that is involved is the solvent, the features of the sequence, and physics, right? Can I predict that? Why not? So I'm looking at you because you shake your head the most.
I don't know.
So John Molt, when he at the first CASP meeting in Asilomar, California in December 1994, stood up and just went through the history and showed the first, I'm no longer sure about the numbers, but the first time, and I cannot not remember which journal, but one of the first journals was Washington Post that had a publication of the protein structure prediction problem has been solved. And the first time this was in the public press was in the 60s. And the second time it was solved was in the 70s. The third time it was solved in the 80s. And actually the CASP had just been after the next time that somebody had published, that some public newspaper, the New York Times in that particular case, had published that the problem had been solved. The problem was you cannot solve the same problem 4 times in history, every decade or something. That already indicates that something is wrong. And what is wrong is that people worked on one particular protein structure and they had methods that reproduced that structure, but didn't work for another one. Now, those were not people who cheated. Those were people who sort of became victims of over-optimization. And that, in fact, is the major thing that you have to be careful with in computational biology. Now, John Molt's idea was we should find a way as a community to safeguard against that, to sort of internally assess how well we do. How can we do that? Well, let's just connect to the structural community, the people who do structures. Let's say we begin our experiment in March this year. Then I will call to all my friends who are experimentally determine structures and find out who will determine the structure over the summer. That sequence I will take, put on a server, and everybody who believes they have a method to predict protein structure, they can predict for that sequence. Over the course of the summer, the publications will come in. Whenever the paper is published, there will be a closure. So nobody's allowed to predict anymore at that point. Then we will collect all these predictions at the end of the summer. That is done by the CASP assessors. That's the term here, CASP assessor. They will look at the predictions, and then we will all meet in December, and we'll see how well we do. That's CASP. And that has been done since 1994, every 2 years. Over 55 publications, 1 with over 800 quotes, 10 with over 100 quotes, H index of 27. Sort of okay, but not very impressive. Now, there are these Sippel potentials. So the moment, some of you may have heard of that, when your name is associated with something that is a theme, then you know that this guy did something, right? Knowledge-based potentials. So that is entirely his idea. Or he pushed it through those knowledge-based potentials. So underneath, whatever we can do today in comparative modeling, in structure prediction, is in fact based on something like knowledge-based potentials. It goes back to Manfred Sippel, although it's not always quoting him. But anyway, so COP's classification of protein structures, the idea now is, SCOP is good, but what if Mozen and the bus have a meeting? So we need something that is sort of more quantitative. Manfred Sippel is more extreme. He says, okay, you know, any expert can say anything, but I want a machine to tell me. I want some objective criterion. Okay? So how can we do that? A quantitative SCOP. How can we reproduce SCOP and do it completely based only on comparisons that are automated? And that is the underneath this machinery. That's what they build up in COPs. And COPs starts with a metric. And the metric, so underneath here is the following issue. And Manfred is much better in showing nice pictures about it, and I should have done that too, I'm sorry. So an alignment between two proteins has some similarities in two protein structures. Now assume that I have 60 residues, so I have two proteins that are 150 residues long, and I can align 60 residues at 2 angstrom. So the trace, the C alpha trace, is so similar that all the distances sum up to less than 2 angstrom. That's one. And in another one, I have 45 that add up to 1.5 angstrom, which is better. Right? Think about this problem in a way that you clearly, you may feel you clearly know the answer to that, but then I rephrase it in a way that you clearly will no longer know the answer. We cannot tell. Let's look at a different problem here. A is similar, this means is similar to B. B is similar to C. What about A and C?
We can't say.
We can't say. And that's what the metric is about. A metric, a mathematical metric, as you know, is such that you can in fact infer that then, if that is the case, A and C are similar. But in proteins you cannot do that, because it could well be true that A is this, B is this. Oh, no, that cannot be true. So that's B. Wait a minute, how do I have to construct that?
That's fine. If the upper one is B.
No, but in this case B and C are not similar.
They are.
But I have to clearly call this B, and that A, and that C. Back. So in this particular case, I have B and C similar, B and A similar, but C and A absolutely not similar in this construction. Now, the idea here is to base COPs on a principle that in fact removes this asymmetry, that introduces a metric into the comparison of 3D structures. The formulas are here, is seemingly a simple step, but it's something that nobody in the field had ever been able to do. It's an ad hoc way of doing it. It's a simple bioinformatics publication that again I don't quote. I'm sorry, Manfred. No. But it's a brilliant idea. And that is in fact the entire concept behind coming up with a quantified SCOP. That's what you need. That ends all the judgment calls. Now you have a criterion that you objectively can compare. You may argue that there are many things amiss, but now you can really measure. And in fact, what they do measure is you get very far. This one here looks at the, I'm wondering because you looked at the time. So we have another 20 minutes, right? And we still have a few slides. So this looks at the date here, number of domains, and it looks at the level of novelty. Zero novelty is blue. So essentially it means that the structure that has been determined anew in that week, or the set of structures, in fact it's nothing that you could not have trivially done by comparative modeling. Okay, and now sort of we get down here, black is completely new in some sense. And you see there is not a lot of black. Half of it had something that is new as yellow. Find the yellow, right? On my quality. Okay, now let's get into the green. In the green area between 1/3 and half. So that's green. Okay? So that's the number of things added to the PDB. And in a very, very, no. And in fact, most is a repetition of something that we already have. Most is on the level of, it in some sense could have been done by comparative modeling. That is what this method allows you to do in a large scale and look through it. The method has other advantages that then you can go into the top search engine, look at it, play with it, that allow you to in fact operate on larger units. That is the only framework that allows you to in fact look at protein interactions in a structural sense. So you can now look at protein A interacting with protein B and do a structural comparison for that pair of proteins. Again, there's no other site in the world that can do that. Play around with it when you want to compare protein 3D structures. COPs, not widely used. Most people today use CATH, but clearly the most objective and in some sense future-leading database out there. How do we compare 3D structures? And most of the slides that I use here are from Pastoris Köll, UC Davis. Again, so we begin, we have to, in the first step, identify equivalent positions, so residues that match in the 3D structure, and then find a superposition. So take here the helix out, and find a way to put it back in, meaning, so where do you put it? And then align it, okay? I talked here, I had this issue of the 2 angstrom, 1.5 angstrom. That in fact is the number. So the difference in the backbone atoms, the main chain difference, is the RMSD, the square over the differences. Again, this one here has 2.8, this one has 2.82. Those are different fragments. Do they look equally similar to you?
They correspond to different parts of the green one.
Yes.
So yes, they are pretty similar.
They also have...
They're not identical, by the way. Be careful. Yeah. The alpha helix, what were you about to say?
There's like a kick and the green one at the bottom.
Oh no, that, yeah, but oh, yes.
Just cut off.
1, 2, 3, 4, 5. 1, 2, 3, 4, 5. Yeah, you're right. The kink is in fact in the helix. You're right. That's true. So that is most likely not counted here. But the other observation is going back to a similar problem here. What I show you is the sum. This region here is longer. So that fits, in fact, most likely much better. I briefly mentioned Willie Taylor. Joel Sussman, by the way, was one of the people who, Joel Sussman started the PDB and was one of the people who was behind this predict the protein or the organism from the amino acid composition. That's Chen Kezar. Christine and Willie developed the first structural alignment algorithm, SCOP, sequential structural alignment, that essentially uses beta distance matrix and applies a double dynamic programming. I will talk about dynamic programming. I will not talk about double dynamic programming. Double dynamic programming, you have heard double dynamic programming. It's just twice the same. It's very simple. It's not very often applied. There's one person in the world who understands it. It's this one here. There's one person in the world who has applied it. That's this one. And for everybody else, it's just one of those things you can do. But the point is that the idea here is you want to align 2D maps. So you have 2D maps and you want to superpose them. You cannot do that by a straight dynamic programming. That's why for this problem you clearly need it. So essentially here, what you have at the residue, an environment. Okay? An environment and distances for the environment. And you essentially want to align the environmental distances of these two. I'm not going to go into it. You optimize a slightly different function, but essentially it's still the same thing. You condense the, or you want to make these 2D maps look as similar as possible. The problem here is you lose directionality in the 2D map. You replace distances by interatomic vectors. That is a matter of speed. You include sequence information, day of type in the olden days. That's simply done to speed it up again, to not too often try things that are different. Here is the same idea, but more, in some sense, more extreme. So while the SAP algorithm looks like 2D comparisons and uses sequence, this completely throws sequence out and only looks at, it's called DALI, for distance alignment something. Oh, I'm sorry. Published in '93, Lisa Holm and Chris Sander. Lisa is now at the University of Helsinki. Chris is in Sloan Kettering in New York. And again, the idea is you look at these two distance maps and you cut out areas in these distance maps and you compare, you slide these areas through and compare these. You find all the possible comparisons by Monte Carlo, so by simulation. Here is one that comes from Munich, Voro Line, Fabian Birtzle and Ralf Zimmer, you all know. Here the idea is simply you look at local environments, graph environments, and then align those against each other. That advantage here is once you get it to the Voronoi trees or Voronoi graphs, you in fact can map it to a simple dynamic programming. That's the great aspect of that. It's very fast, it works very well. But I do not know that many people use it except for the authors. Now, here we have this typical kind of example. You sort of jumped on it in the kink of the helix. We have two forms of calcium-bound calmodulin. That's the ligand-free form, and that's a complex form. That's the same thing. Doing the same thing, same function, same structure, doing something similar. But it's not trivial to see the similarity. In fact, by whatever I showed you so far, they are utterly different, these two shapes. And that is one of the issues. One way in which you can get rid of that problem is by, rather than having the global alignment here, which is really dismal, and that again is a slide from Patrice Köhl, you look at local regions, so you cut it out. And then you see that you can essentially, the helices binding the calmodulin, the finger, that you can immediately see aligns very well. But that brings us into the problem that then, how many cuts can you do? And ultimately, this brings you back to the issue of what is the unit? Are we going to cut it down to the level of a single helix? Or, as in this case before, even fragments of helices? Where do we stop? That requires in itself, the answer to that question requires that you have a model underneath that tells you, is that better here? 60 residues at 2-point angstrom? Or 45 at 1.5 angstrom? And you were all happy not to answer that question, what is better? So there is no simple model for that. So it's a judgment call. Here's another problem, okay? There are tons of structural alignment methods out there. Here, just a few. In fact, that few comes from Rachel Kolotny, Patrice Köhl, and Michael Levitt. Publication in 2004 from Rachel in JMB, in which Rachel compared different structural alignment methods and found that fraction of false positive, fraction of true positives, the background I believe was SCOP, I can't remember, and those are in fact different alignment methods, right? So one take-home message, they differ. One take-home message, the gray here, that is this one here, the best of all is this one here. So if you would know what the right answer is, and you would take all the alignment methods and pick the one that is right, then you will get to this line here. That's as good as you can. But no method gets anywhere near it, is essentially the news. So what is the best method then? And so Rachel, so she clearly is the #1 expert in the world to answer that question, and she says, I don't know. So what do you want me to do? Use them all? Just do the right thing, she says. So conclusion is this is a very simple problem, and you don't know what you do. My suggestion: look up CATH and SCOP. Those give you alignments immediately. That's one way. Another way of doing it is you run the modeling program that gives you another alignment. We will get to that once I talk about comparative modeling. And the third answer is use the stuff that I showed you before, COPS, QSCOP, and the top search from the Zippel group, which is in fact not on this one here because it was not published at that point. That's my best bet. The trouble is, just like MD in molecular dynamics, this is still not automated. Okay. Again, we will get into alignment methods. Oops. Yes, exactly. Next Tuesday, and then we will get into 2 skips, the May Day and the student assembly, and then continue with alignment on the 8th of May. Any question? So one question, not everybody was in the room, when the examiner will be, and that is to be discussed with Lothar Richter. It seems that there was a majority on the answer of Tuesday in the exercise time. But again, clear that up in the next exercise. Any other question? Thank you for your attention.